So what are we going to do in this lecture? We're going to talk about different pathways through which pain is going to be transmitted to the brain and modulated. And the reason that we're going to be looking at that isn't just because I think it's insanely fascinating, and I know you will too, but also because that's where we're going to identify where the different drug targets are. And we're going to see what drugs can work where. So the first thing that we're going to talk about is inputs and the spinal central synapse. And we have nerves that are pain-transmitting nerves, and those are primary afferents that are sending pain signals from the site of pain into the spinal cord. We're going to have a central synapse occurring in the spinal cord here, where that pain nerve is going to transmit to an ascending nerve that's going to send information to multiple parts of the brain. And we're going to talk about all of these in more detail in the next slides. And then we're going to also have an ability to modulate pain, because we don't want to just have pain that the body is not able to ignore, or not enough pain and then not modulate our behavior and not stop doing something that's going to harm us because we're not feeling pain. So the body has to have a way to turn up and down pain signals. So we have another pathway, which is from the brainstem, goes down to the spinal synapse, and is an inhibiting pathway. And so that's going to go down and stop pain transmission across that spinal synapse. So to talk about these in a little bit more detail, we're going to start with those inputs, and we're going to start with the spinal central synapse. So pain transmission at the level before it goes to the brain is called nociception. And that's to distinguish just this purely sensory stimulation from what happens once it reaches the brain, which is we actually feel bad, we feel negative about it, and that's going to be when we call it pain. So that pure sensory transmission is called nociception, and it's going to start with some kind of stimulus. So in this case, I am going to smack your hand with a hammer. That's not a hammer, that looks like something else, but it's a hammer. So I'm going to smack your hand with it, and that's going to create some kind of stimulus. And we call any painful or damaging stimulus noxious. So we've got this noxious input. The nociceptive fibers, which are the neurons that carry pain signals, are going to have free nerve endings here. What free nerve ending means that it doesn't have a synapse. So there's no other neuron that's coming onto it and giving it signals to activate. So there's various things that are going to activate that receptor. But they have receptors, and they have receptors for other stimuli, which we'll talk about. So whatever this noxious stimulus is going to activate those nociceptors, which are the free nerve terminals. That's going to send a signal along this nociceptive peripheral nerve, and we call that also a primary afferent. And that is going, here's the cell body, we're going to send an action potential along using sodium and potassium channels. That's going to go into the spinal central synapse, and then that's going to synapse onto, so here's our synaptic terminal. It's going to synapse onto those neurons that go up to the brain. We're going to talk about that spinal central synapse in the next slide, but I want to also explain a little bit about these nociceptive peripheral nerves. So I say here we have different types of those, and that's because we have to have different types of peripheral nerves. Each type has a different signal. So we have touch, we have temperature, we have itch, and we have pain, and we have different variations of those. So each of those types of sensations is going to be transmitted through a different type of neuron. So we've got what we call C fibers, and those are going to be a lot of the pain we talk about in chronic pain conditions. We've also got what we call A delta fibers, and I'm just throwing it right here, but you can look at it coming from the thumb, here in this case, into the spinal cord. So we've got A delta fibers and we've got A beta fibers. And A delta fibers are another kind of pain neuron, and those are more sharp pain. And then A beta fibers are just regular non-painful touch. We're not going to be worried a lot about the touch. We're going to be looking at the pain neurons, and we're going to just basically focus on the C fibers, because those are going to be the ones that are usually activated when it comes to chronic pain. So when this fiber goes into, and I'm saying fiber, but that's really what we're calling a neuron, this sensory neuron. It's going to come into the spinal cord. And like any neuronal transmission, it's got to activate those presynaptic voltage-gated calcium channels. And then that's going to initiate the release of neurotransmitter. And in this spinal central synapse, we're going to see glutamate, substance P as a neuromodulator peptide, and we're going to see CGRP as a neuromodulator peptide. And substance P is substance P, and CGRP is calcitonin gene-related peptide, but you don't need to know that. So we're going to look at this actual spinal synapse in the next slide, but first we're going to look here at that free nerve terminal and how that pain neuron actually gets activated to begin with. So if we come down here, we're going to see that I have the free nerve terminal drawn here. And when that's activated, those signals are going to go, that depolarization is going to go through the dendrite to the cell body and then to an axon. So in order to be activated, we still have receptors, and we call them that even if they're not being activated by a neurotransmitter. So for every type of neuron of the sensation, we're going to have a different type of receptor. I'm going to draw them all on this same nerve terminal, but they're not. They're going to be each on a different type of nerve. So we've got receptors that are going to be activated by pressure. We've got ones that are going to be activated by various different inflammatory mediators. We've got receptors going to be activated by heat. One of those are called TRPV1, and we're going to see those again. We've got receptors that are activated when they are exposed to ATP, which should not be outside of the cell, so they shouldn't be exposed to that normally. Ones that are activated by acid, so low pH, and ones that are activated by histamine. So these histamine ones are going to be usually on itch nerves. We've got these TRPV1 that are activated under high temperatures, so they're activated for heat. So how are we going to activate some of these? Well, obviously, pressure and heat are going to be activated naturally, but how do we get some of these other activators? Well, up here at the top, you can see here's just some tissue cells, some random tissue cells that if they're injured, you get cut, if they get smashed, when those break open, the intracellular contents are going to leak out. And those are going to include hydrogen ions, they're going to include ATP, and those things really should not be at significant concentrations in the extracellular fluid. So as soon as this nociceptive nerve ending sees ATP, and that binds to this ATP receptor, that's a signal that there's been some tissue damage. So that's going to activate this pain nerve. We also have glial cells and any histamine-releasing cells that are going to act then down at the histamine receptors. So that's going to show us itch. And then all these inflammatory mediators I mentioned are going to be released in multiple different ways. So they can be released from glial cells. They can be released from vasodilation. This is a very poorly drawn blood vessel. So vasodilation and glial cells can release a lot of inflammatory mediators. They're going to include things like interleukins, prostaglandins, thromboxanes, and various other types. So now we can talk about that signal when it reaches the spinal cord. And we're going to reach the spinal cord in here, and that synapse is going to take that primary afferent nociceptive neuron. In this case, it was a C fiber. And it's going to go across that central spinal synapse and then go up the ascending pathways into the brain. So this is a really simplified diagram of that spinal synapse. And you can kind of see here that I am not going into the type of detail that is actually in that synapse. There's some excitatory interneurons. There's inhibitory interneurons. There's a lot of back and forth modulation between the cells, but we're just going to show it as a straight synapse here. So that stimulus came in, we had the nerve terminal with nociceptive receptors in the last slide, we had the peripheral nerve that came in, and now we're going to talk about how that synapse works. So we had the action potential come that activated voltage-gated calcium channels, and like with all other nerve transmission, that's going to cause neurotransmitter release. We really talked about neurons as having one neurotransmitter, and for a long time we thought that was always true. They could only have one. Now we know that some neurons actually have more than one neurotransmitter, but it's also not uncommon for neurons to have a major neurotransmitter and then some peptides, like inflammatory mediators, that can also be released. So in this synapse, which is that major synapse in the spinal cord, we release glutamate. That's our major neurotransmitter. But we also release substance P, and we also release CGRP. Those are going to cross the synapse, and glutamate is going to act at NMDA receptors, which were those ligand-gated ion channels. It's going to act at AMPA receptors, which are also ligand-gated ion channels. The CGRP is going to act at CGRP receptors, and the substance P is going to act at TRPV1 receptors, which are ion channels. So when we fire this neuron, we're going to get glutamate binding to the AMPA receptor, CGRP binding, TRPV receptors activated from substance P binding, and we're going to bind to NMDA receptors, but they're not going to really be activated, because when we talked about NMDA receptors before, they have a magnesium block, and that magnesium only moves out of the way after that neuron has been highly depolarized. So under normal pain transmission, you have a low to moderate amount of signaling. The AMPA receptors are going to depolarize, and the TRPV receptors are going to depolarize, and the CGRP receptors have different kinase effects. And yes, it's going to send activating signals, but usually it's not going to be depolarized enough to activate the NMDA receptors. And that comes into play later when we get chronic pain conditions, when we do activate NMDA receptors. So that's the main transmission there. And so you can see we're going to have some drug targets at CGRP, at substance P, at glutamate transmission, at voltage-gated calcium channels. And then we're also going to have a couple of additional factors here, because the brain and the nervous system cannot make something simple. It can't just have one pathway. It has to be able to modulate it. So in this case, we're also going to have this descending inhibition that was that serotonin and norepinephrine pathways that are going to be able to come, and they're going to be able to decrease release and decrease activation. So those pathways are going to inhibit transmission. And we've also got opioid receptors on both of these sides, so that opioids, when we give them, can also inhibit transmission.